The overall goal of this stencil-based micropatterning method for human pluripotent stem cells is to provide a simple and robust means to generate spatial environmental gradients that control stem cell differentiation patterns.
This method can help to answer key questions in tissue patterning using a human stem cell model, such as how spatial gradients in cell-cell and cell matrix adhesion forces can help to organize stem cell differentiation fates. The advantage of this method is that it does not require additional surface modification to generate cell-adhesive ECM patterns for cells to attach to. This means the method is compatible with various ECM and substrate configurations specific for each cell line.
Visual demonstration of this method is critical as the micropatterning steps are difficult to learn because it requires precise and careful handling of the stencil and the cells.
For this procedure, the PDMS stencils must already be designed and fabricated. Likewise, have human ESC cells growing on basement membrane matrix such that the colonies will be ready for seeding later in the procedure. To begin, load a dish with 700 microliters of 70% analytical grade ethanol in ultra-pure water. Then, place the PDMS stencil on the solution and set the dish into a biosafety cabinet to dry out overnight. The next day, check that no bubbles have formed under the stencil. Then, seal the dish until the cells can be seeded onto the stencil. Prepare an aliquot of human ESC quality basement membrane matrix by adding 16.7 milliliters of DMEM F12 to make a 1.5x solution and keep it on ice.